Hey guys, and welcome back to the ASX Investor channel, and welcome to a very exciting interview. Today, we're speaking to a really esteemed expert in the field, and we'll be diving into the world of biotech. We're really excited and grateful to be joined by the San Giacomo Family Chair in Surgical Oncology and the Chair of the City of Hope Department of Surgery, Professor Yuman Fong. Professor Yuman Fong has got an incredible history. He served for over 20 years at the Memorial Sloan Kettering Cancer Center. He's renowned for his extensive work in the field of liver and pancreatic surgery. And he's also the inventor of the CF33 Oncolytic Virus. The oncolytic virus has been licensed by Imugene, ASX IMU. As you would be familiar with the story, we've previously spoken to the Imugene CEO and Managing Director, Leslie Chong, on the channel. And so today we'll be talking about the CF33 oncolytic virus on an exciting day with the first patient being dosed. And we'll be talking about the story and the opportunity for truly what it could mean to make a big difference for those affected by cancer around the world. Professor Fong, it's an absolute pleasure. Thank you so much for your time and welcome to the channel.
Oh, thank you very much for your time and your interest.
There was so many different places that we could have started this interview. I was thinking about where to begin, and I thought an interesting place might actually be with a bit of your background, from a BA in medieval literature from Brown University through to an incredible career in the medical field, obviously a range of different factors. I'd love just to have a bit of an idea about what's your background, if you can share that with your audience, and most importantly, what's led you to today and having your passion within this field.
Oh, listen, I ever since growing up here in America, I've wanted to be a physician. I wanted to be a doctor and a surgeon. And so I was very fortunate in when I left high school here in America of having been given a spot in medical school. And that's because in America, usually you go to university and then you go to medical school. But I was given a spot in a 7-year medical program here in the United States at Brown University. Because of that, my advisors said, "You're going to do science your entire life. Why don't you go do something that may be of interest that in the first few years?" And that's why I studied poetry, because it was always of interest to me, literature, poetry, and how epic poets remember the thousands and thousands of lines of epic poetry. But I've always wanted to work in research. Throughout medical school, it was very clear that I was going to pursue a career in surgery. But as part of the surgery, academic surgery, I wanted to go do research. And I was very fortunate to have fallen in with a group of researchers that discovered cytokines. What cytokines are really the proteins that are triggered by our immune system to either fight disease or cause disease. And I was part of the group that discovered a very important protein called tumor necrosis factor, and discovered that it was the most important protein when patients humans get sick from infection. And from there, it was really a 30-year journey in studying how the immune system works in disease and in health. And since I was a cancer surgeon, it was just natural that I would be working on things to try to enhance human immunity to try to fight cancer.
Yeah, I really appreciate that overview, and it's incredible to hear about the journey as well. Of course, that's led you to, or one component of that is the oncolytic viruses. I guess for the viewers who may not be as familiar with the oncolytic virus, it's a virus that kills cancer. It likes and almost exclusively infects cancer. It'd be great to give that overview about what is an oncolytic virus, the overview of the field. There's only been one approved so far, CHECKvacc, and why you're so interested. Why is it such an area of hot interest for the broader sector?
Well, it's been known for a long time that viruses can kill cancer. And for over 50 years, people have been trying to find a virus that kills cancer well enough to use as therapy. It's not far-fetched to think that we can find such a cancer and design, it's not far-fetched that we can actually find such a natural virus that only kills cancer, or make a virus that only kills cancer. Because if you think about it, in hepatitis viruses only infect liver cells. In meningitis, the viruses only infect brain or neural cells. And so the idea that there is a virus out there, or we can design such a virus to only infect cancer, not far-fetched, and people have been trying to do it for many years. And I think we're very close to having a virus that does just that.
And I guess what I found very interesting about the way that you tried to approach it, it was quite an industrial process. You took a range of different vaccine strains with the Vaxinia virus, essentially put them into cancer cells. And the way that I heard you describe it was let nature to do its work. So then put it through high throughput screening on the NCI60 and just assess which were the most efficacious and which was able to kill cancer best. Can you give that overview about how you went through that process and ultimately how CF33 was born and why you're so excited about it?
So what we did was this. There are a number of viruses that have been given to man and have found very safe as vaccines. We said, what if we took a whole bunch of these? And in this case, we took 9 different viruses, put them into the same cell. Viruses live to trade in genetic materials. It's just that in nature, it's very rare that more than one virus will enter a single cell. And so having 9 different vaccine strain of viruses in a cancer cell, these viruses traded all kinds of genetic material. So we isolated 200 different new viruses that never existed before. And then we went through and said, why don't we just test them against every good cell line we know that we want to kill. And so we took something called the NCI60. NCI stands for the National Cancer Institute. This is the 60 cell lines that is used by the NCI to screen for any new cancer therapy that's coming through to see what cancers it might be good for. And I said, I don't care about just a virus that kills only brain cancer or only pancreatic cancer. What if we just said, we only care about those new viruses that killed every cancer? And we were able to find 5 or 6 that were really good at this. And then we said, how many of these are really safe for animals and potentially for man? And that's how CF33 was born. This is a virus that kills everything in the NCI60 and was found to be very, very safe when we injected it in the animals. So we think we have a virus that is very close to one that only infects cancer.
And just to drill down a little bit on that. So I think the numbers were 97%. You had the ability to kill cancer. Can you give that context to the viewers? What generally would be enough to progress it through and continue on? Like how many of the different cell lines would generally people be seeking?
So when people are designing a cancer killing agent or a cancer therapy, if the therapy is really good at a single kind of cancer, that's usually good enough. Okay, people are generally saying, let's go cure leukemia or let's go cure pancreatic cancer. And but what we are actually trying to do is to find something that could kill many, many different types of cancer. And that's because it's been very clear over the last 2 decades that in order for our immune system to attack cancer, we actually have to have the right immune cells at the local area. We actually have to be able to activate them to recognize the cancer as foreign, and viruses are really good at that. And therefore, trying to find one that could be used in many different kinds of cancer for such usage, that's what we're trying to do. And now we get to put it to human testing, and it's a very exciting day.
It's a very exciting day, of course. It's such a milestone day. When we're thinking about some of those benefits, before we drill down, of course, into the announcement, I would be fascinated to hear, of course, you've got that ability across a large range of cancer types, but it's also effective at extremely low dose, good utility for combinations. What are those other benefits and the ability that you think to continue to scale this moving forward, depending what we see with the trials?
One of the things that we noticed early on in testing CF33 was how remarkably potent it was against cancer, meaning that in many animal models, we were able to cure animals of cancer with as little as 1,000 particles of virus. Let me put that in perspective. In the last generations of viruses that were being tested, it was very rare that if you can impact upon the outcomes of the animals with anything less than 1 million particles or 100 million or even 1 billion particles. And now we're down to 1,000 for this current virus. And we didn't see any toxicity in animals until up to about 1 billion particles. And therefore, huge window of what is potent for cancer and what is safe for man or animals. And so quite excited about it.
And that, of course, then brings us to the news today. It's such an incredible day. CF33 with the first patient dose. So I'd love to hear a couple of different reflections. Firstly, how it feels to see your life's work now into the clinic, and then also what you're hoping to see with these trials now underway.
Oh, very excited. So this is our flagship virus that has gone to man today. But in reality, we have a version of it that's already gone to man and just a really terrible disease called triple negative breast cancer. before the virus was licensed out to Imugene, I had already started down the path of treating triple negative breast cancer because it's such a terrible cancer. It's a cancer that does not, it is a breast cancer that does not respond to hormonal therapy. It doesn't really care about things that block estrogen or block HER2. And therefore, most patients who get it, I have it recur. And when it recurs, generally patients die very quickly. It's a great disease of need. And so a different version of CF33 we had already put into that disease. This is a version that actually also puts an anti-checkpoint inhibitor right in the site of the cancer. And the good news there is that we're already well into that trial, and the agent is very promising. Okay, but what is great today is that we've now taken the parental virus, and we're going to now try to direct it against many different kinds of cancer. We're also going to test it by injecting it intravenously, which is like the holy grail of viral therapy, because the last generation viruses that were tested in the last 25 years, most of them could not be injected intravenously to effect. We have very good data in animal work that our virus at CF33 should have a good chance of working intravenously. And if that's so, we are likely to be able to go deliver it for many, many different kinds of cancer in many different settings. And so again, but first steps first today, really great day.
I can see the passion where you speak about the opportunity to really make that tangible difference for so many out there. So I'd love to hear, so from here, this is the first step, this is today. What are we looking forward to now in terms of the process moving forward?
This trial is being set up as a multi-center clinical trial. The reason for that is because we want to move with some speed, and we want to be able to enter many different patients with many different kinds of cancer. We also want it verified by many different investigators, and so again, we want to move forward, get data that says safe, then get data that says which cancers in humans can we actually have an effect. Then it's also been very clear that there are many other cancer treatments out there that require just turning on the local environment of the tumor to be much more immunogenic. And we think we're going to be able to partner with all of those agents to make them better and to make us more relevant. So therefore, we have already taken many steps forward in setting up partnerships with checkpoint inhibitors, with CAR T cells, with other kinds of immunotherapies. And where I'm hoping that as soon as we get some data of safety and efficacy in current trial, we'll be off in partnership trials that will tackle some of the toughest cancers that we've not been able to treat, and make a great impact on human health and treatments of cancer.
It really is so exciting. I know that everyone's obviously got a story of someone around them that's been affected by cancer. Drilling down into some of those combinations, as you mentioned before, CHECKvacc, it's into the clinic now, armed with a PD-L1. Can you speak about some of those early indications or what we're seeing at this stage?
We are actually restricted by the FDA to talk about the data until it's fully analyzed. Okay, but there's no doubt the trials are going along, and it's proven to be very safe. And we are getting biologic readouts that are relevant and making us very excited about where the future of this agent and this platform is. Because if you think about CF33, it's not a single agent. It is the entire viral platform that allows us to deliver not only the virus, but any gene, transgene that's carried by the virus to go stimulate the local immune milieu to go and fight cancer. And then ongoing, we also have a very exciting next agent where we are planning on putting right on top of tumor cells targets for CAR T cells and other agents. And so again, we're also embarking upon a combined virus plus CAR T strategy that we're hoping will be in man very shortly.
That onCARlytics there, I guess it's worth drilling down into it. It's often called as well the holy grail of treatment, getting that ability to treat the solid tumors potentially. Yeah, I'd love just to hear about why that's so exciting and so important as well. We know that blood tumors can be treated, but solid tumors is really that significant opportunity there.
CAR T cells are a very exciting aspect of immunotherapy. And that's because what it is it's taking a human being's own immune cells out training them to recognize a protein on cancer, sending it back after it's been activated to kill the cancer. And so it's been highly effective in liquid, what we call liquid tumors. Those are the leukemias and lymphomas, because there's a protein called CD19 that sits on top of a kind of cell called B cells. Those are the cells that normally make antibodies for us in our bodies. So when somebody gets a B cell lymphoma, B cell leukemia, now we are able to give CAR T cells to rescue them even after they have failed chemotherapy, after they have failed bone marrow transplant. And so very exciting that there are 3 different CAR T cells that are already approved for CD19 positive liquid malignancies. Finding a CAR T cell that will attack solid tumors, meaning things like lung cancer and breast cancer and colon cancer, has been much, much harder. That's because most of the things we can attack on a solid tumor exist in normal tissue. And therefore the off-target effects of that. So when we go attack CEA, which is a very common protein in cancer, in colon cancer and in lung cancer, but unfortunately it's also found in normal intestines and other normal tissues, the CAR T cells can hurt those other normal tissues. Since our virus, CF33, is so good at infecting cancers and only infecting cancer, we have designed a version of it that we call onCARlytics. This particular virus not only infects the cancer, starts killing it, attracts immune cells, but also puts on any cancer this protein CD19. And suddenly, everything that we designed for CD19 therapy, including CAR Ts, including antibody therapies against CD19, could potentially be a secondary agent that partners with CF33 for killing solid tumors. So this is kind of what we call our solid to liquid program that we hope will become a successful combination therapy of oncolytic viruses plus CAR Ts or other agents that target the CD19 protein.
It's an incredibly fascinating story. Looking forward to potentially having a discussion as well when that gets into the clinic as well at some stage. I'd love to ask you too, of course, you've spent an incredible amount of time developing CF33. How did you go about the decision to partner with Imugene? Obviously, you've developed a great relationship with Paul Hopper and Leslie Chong. What was that decision and why is that relationship so fruitful as well?
Well, I was working very, very hard in academia to try to move all this stuff forward. And then I met Paul Hopper. And it was very, very clear. He was experienced. He was, and he was someone you can talk to and partner with. Okay. And then his record with working with a previous virus company that was able to be moved forward into the commercial space was also very attractive. Since starting to work with Paul, he's brought in this entire team of Imugene and Leslie Chong and every step along the way has been a true partnership and a professional partnership. So I have utmost confidence, not only in their ability to do the business aspects and the trials aspects of the work, but to be able to truly work through the important logistics of getting this through human testing as quickly as possible.
It is a truly milestone day. And as we can see just from the discussion, it looks like there's plenty more potential milestone days up ahead in the future. I'd love just to leave the final question with you. Any final reflections that you had for viewers who might be new to the story or those who might have been following the story of CF33 and Imugene over the past few years?
From the Imugene standpoint, the other thing that I've been very impressed with is that as I look at the portfolio of the company, it's not just viruses, it's immunotherapies of many types. And it is a clinical testing stage company that's been highly successful in getting trials done and getting results out. And so very, very excited about this startup that is more than a startup. Okay. And but from the standpoint of viruses, there are so many promising steps forward and directions forward for oncolytic viruses. And since we believe we have one of the very best that's ever been designed, so quite excited about what the future holds and looking forward to many more trials moving forward, but really in reality looking forward to seeing successes that translate into patients living longer, patients being cured from cancer, patients spending more time with their families. That's what we're really, really looking out for.
That is the CF33 story. If you are interested in learning more about Imugene, we'll provide a link to some of our past interviews with the CEO, Leslie Chong there. Professor Yuman Fong, just want to say big congratulations for what truly is a significant and milestone day, but ultimately at the end of it, we'll continue to watch and all be supporting, hopefully making that difference for many around the world. Thank you so much for joining us.
Thank you very much.